Hello, it's Ellen again. You're watching Scientifica Mathematica, and as I promised, I'm back with my second video on elaboration of genetics and heredity. Because, as I said, if you watched my previous video, it's my, I guess, specialty. It's what I'm passionate about in everything about biology, although I love biology as a whole. And so, genetics, there's some things you need to know. It's not just genes, it's not just simply the passing of traits, although that's basically what it is. You have something in your body, known as DNA. It looks something like this. And there's all these little. And it looks something like this when stretched out. It's a little messy, I'm sorry. But anyway, it's a twisted strand. Two strands twisted together, and it looks somewhat like a ladder. And basically, what it is it's the code for who you are. It literally codes you. You're not a robot or anything, but think of it like that. It's called deoxyribonucleic acid. Let me spell that out for you. Deoxyribonucleic acid, also known as DNA. And this is very important because it's what makes you look like your parents. It's what gives you your dad's eyes or your mom's nose or your grandma's skin color or curly hair or something like that. But it's all this deoxyribonucleic acid is exactly what genes are. It's what makes up your genes. And it's really small, you can't see it. So this is just approximately what it looks like uncoiled and really large. And so before we start, I'm going to go over some vocabulary. Whoops. Oh. Okay. So the first word you need to know. Genotype. Your genotype is simply your genetic makeup. Your genes, your DNA, what's in there. Your alleles, which I'll explain later. Your traits, your genes, everything. And phenotype is actually quite the opposite. It's the physical makeup. That does not fit. Your physical makeup. Let's make that a little bigger. The physical makeup of you. Like your traits. The ones you can see. Like how your skin is a nice shade of tan or very pale, or how your eyes are blue or brown or green. That's all phenotype. So the next thing you need to know is we call it a Punnett square, I guess. It's created by this dude called Reginald Punnett, which explains why it's called Punnett square. And it's used to predict the traits that are present in offspring, both phenotype and genotype. That's the power of this thing. It's basically really simple math, actually. And it looks something like this. It's usually a square. Has a couple of lines dividing it. Oh, whoop. It usually looks something like this line there and a line there. And that's what a Punnett square is. It's usually, this is a monohybrid cross. Mono meaning one because it's one. And I'm going to explain what exactly a Punnett square is and does after we go through this vocab. So you have that. Next, you have an allele, like I mentioned earlier. It's known as an allele. And it's one of the chromosomes in a pair. And like I said, your DNA is double. There's two strands. Whoops, I spelled allele wrong. Your DNA is made up of two strands. And in reality, those little ladders that connecting it, those little rungs of the ladder you see, they're actually made up of two different parts, split in half. Or not exactly half, but two different parts. And it's one of those chromosomes. There's 4 types. What an allele. Next thing you need to know is an allele. Like I said earlier, it's the alternative form of a certain trait or gene. And we usually represent them by letters. For example, we can use here an S. That might be one allele. And sometimes, actually all the time, there's another. And capital and lowercase usually means something different. For example, if I were to lowercase, it means something completely different. But that's exactly what comes next. There's something called a dominant trait. Dominant trait means it's dominant. It masks another allele. And there's this thing called the law of dominance. What a creative name. And it states that the expressed trait is always the dominant allele, meaning if you have brown eyes, it's because you either you have the dominant trait allele that makes your eyes brown. There's something called multiple alleles, but I'll explain that later, because what you usually see, like your eyes, it's a singular allele. The next thing is recessive. It's the opposite of dominant, actually. It's spelled like that. And it's an allele that is likely masked by a dominant allele. And the special thing about it is it's only expressed when 2 recessive alleles are present. Because if a dominant allele isn't present, you can't follow that law of dominance. So recessive, for example, and that's what it is. A capital letter typically means it's dominant trait, meaning no matter what, it's expressed until it's not there. You have something called a recessive trait, which is usually notated, what is the word? With a lowercase letter. And so usually your pairs look like this. Let's take for example, if capital S if the dominant trait was long fur, and maybe lowercase s means short fur. Whoops, why is that moving? Oh no. It means short fur. And so if we had that, the animal would have long fur. That's not big enough, is it? Oh well. Just remember that this one's capital. This one's capital. Okay. Anyway. But if we change that, if we had an organism with this genotype, notice I chose genotype because that's what the alleles are. SS, lowercase s. The dominant allele isn't there to mask the recessive allele, which would mean The animal would have short fur. And so we have all that. And then we go into everything here, dominant and recessive, is something we know as Mendelian genetics. It's a mouthful. It's known as Mendelian because there's this dude, his name is Gregor Mendel, he's dead now. But basically, he developed these rules, the dominant and recessive stuff, how dominant, the law of dominance, how dominant alleles always mask the recessive allele, and how recessive alleles only show up, the recessive traits like blue eyes only show up when you have two recessive alleles. That's what Mendelian genetics is. It's everything up there. And Gregor Mendel didn't exactly get everything. There are other things that he kind of missed. We know these as codominance. I spelled that wrong. And incomplete dominance. These are easily messed up because they're terrifyingly similar and confusing. But codominance is codominance usually occurs. Oh shoot. Incomplete dominance and codominance are very similar things. They're both something we know as are things Gregor Mendel did not get. So codominance is when two alleles that are both dominant and they come together and you see a little bit of both, like speckled chickens, like codominance is. These are usually very special cases and don't appear. But, for example, if the dominant allele W meant white fur or white feathers, just pretend it means white feathers. And B means black, like usual. My pen's being funky. Anyway, you have that. They're both capital, meaning they're both dominant. And in the case of a speckled chicken, I don't think I'm getting this wrong, am I? In the case of a speckled chicken, it has both. So if it has the genotype WB, that means both the white and the black shows up, meaning you end up with a white chicken with black spots, which is codominance. It's when two things are both dominant. Incomplete dominance is very similar, as I said, so it's pretty confusing to tell them apart, but after a while you learn. Incomplete dominance is when two dominant alleles blend. They're both dominant, right? So they both have to show because that's the rule of dominance. But for example, you have a flower and it's red and marked by uppercase R. Let's rewrite that. Uppercase R equals red. And W is white, like earlier. Sorry, I'm using a homemade stylus, which means it sucks. Anyway, you have red and white. And this flower has the genotype RW. And you're like, oh no, is it codominance? It has two dominant traits. Would it be like red and white? No, because there's some, you don't have to memorize them, but there are always special cases. These flowers are actually have incomplete dominance when that happens, which means the flower is pink, because red and white makes pink. And anyway, they blend together. And neither of them are truly dominant. And you end up with something in between. And this is something we know as non-Mendelian genetics. Because it doesn't follow Gregor Mendel's laws. And so for these, you can use a Punnett square to figure them out, but they're often confusing because you don't know. So these are non-Mendelian genetics. And there's a few more things you'll need to know. I want to delete. Okay, homozygous and heterozygous. I should explain the etymology first, which makes sense. You see it when people talk about sexuality as in homosexual or heterosexual. This is pretty much the same thing. Homo, the root homo means, let me draw. The root homo usually means same, meaning it's the same. Homozygous would mean when the traits are either both dominant or both recessive. The alleles are identical, meaning it's like SS, both dominant, or you have lowercase s for both, as long as they're the exact same. That's what homozygous means. Heterozygous is the exact opposite, which I'm sure you can figure out. Hetero means different, or, well, two different. It's two different alleles. Means different. Maybe I should type this next time. Anyway, you have that. And for example, if you had one dominant trait and one recessive trait, that's heterozygous because one's capital, or one's dominant, and one's lowercase. It's recessive. And this is pretty much the basic vocabulary you need to know for genetics. So I'm going to go over Punnett squares now. Punnett squares, like I mentioned earlier, they're basically chart. Let's delete that. It's a chart. So you usually have a square, and it's divided into 4 sections. As I said earlier, we know this as a monohybrid. Monohybrid. What's the B doing there? Anyway, monohybrid cross. We know this as a monohybrid because it's one single allele. You're only crossing one. Dihybrid is when you cross 2, and that'll be a separate video because it's much harder. A monohybrid cross is a type of Punnett square. Punnett squares are used to predict the offspring, the traits the offspring will have. And so let's give the parents, I'm going to use T in this case because the capital and lowercase are different. But let's just give 2 random parents with 2 random traits. For example, parent 1 is heterozygous, has capital T, lowercase t. Let's just say the T is for hair. Curly hair is dominant, straight hair is recessive. So that's parent number 1. And because of this, they have curly hair. Let's say parent number 2 has 2 T's. That means 2 recessive genes. That means they have straight hair. And the offspring always has a fair amount of getting a certain trait. And when people reproduce or animals reproduce, they pass off one of their traits. So for example, like they might pass off T, the capital T. They might pass off the curly hair trait. And the other one might pass off the recessive trait, T. And the child would have long hair. I mean, not long hair, curly hair. But what if they passed the other trait down? What if both parents pass down a different trait? Then the child would have straight hair. But you have to figure out exactly how many times you can cross them. And frankly, it's hard to keep it all. So this dude named Reginald Punnett, let me write out his name here. I don't even know his middle name, but we don't need to know that. Reginald Punnett, he made something called the Punnett Square. Yeah, genius. He named it after himself, or someone did at least. Anyway, Reginald Punnett, he made up this thing called a Punnett Square. As I explained earlier, it looks like that. And what we do is we cross the parent's traits, and in turn, we get probability. We get a certain number, like maybe like 3 chances out of 4, 75% of the time, the child would have, I don't know, straight hair. But we don't know that. So what we do is we cross them. We take parent number 1, and we put it here. We separate. We put capital T and lowercase t. We split it in half. And we put one on the top and one on the bottom. Second parent, we split it as well, split the letters, put one T here, or one allele here, and the second one here. And now we have this. If you know how to multiply, it's the same. If you know how to play Battleship, it's the same. Like, you know how you read it like as like A5. That's exactly what's here. You take whatever's here, and you put it into this box. And so when you drag it all in there, you end up with T. You end up with that. And which means the child will have curly hair because it has a dominant trait. And by the law of dominance, the dominant trait always masks the recessive one. And then you do the same thing with this column, with this trait and this one. You look across and you look down, and this one, it's going to be the same. You end up with that genotype. That child will also have curly hair. And you're going to do the same thing again with this one and this one. And this one, you cross it, you get, you get 2 lowercase t's, meaning the child will have straight hair. And another lowercase. Why is this one doing that? And you get another T. And that's really messy, but you know that the child will have straight hair because there's 2 recessive alleles. And what you end up is for all the possibilities of offspring. And so if you look at it, from these 2 parents, we end up with these 4 possibilities for a child. So if they were to have a child, it'd have 1 out of 4 times it will have this genotype. 1 out of 4 times, 1 out of 4 times it will have this genotype. I'm sorry, it's not cooperating. It will have this genotype. Meaning you have that. I can't draw. Something is wrong here. Here. You have that. And another time, your child might have that. So that's another possibility. Next time, your child could have this. And the last time, your child might end up with that. So basically, there are 4. If you have a single allele trait, there are 4 possibilities of a genotype. And so your child usually ends up with one of the 4. And so you'd say But here's the thing. Phenotype is different. Phenotype only cares about the physical traits. So you end up with this, maybe. These top two right here. You might notice they both end up with curly hair because there's that dominant trait that masks the recessive trait, and you end up with curly hair. But there's also two possibilities, like these two, that the child has straight hair. And so we have 2 out of 4 possibilities, or 50%, or half of the time, your child has curly hair, and another half, straight hair. Meaning, you could flip a coin, and it'd have a pretty good shot of determining whether your child has curly or straight hair. And so that's how it works. I want to do another example. This time, I want to... Is there a way to delete, erase? Oh well, anyway. This time, I want you to do it yourself. Tell me in the comments what exactly this ends up being. So, right now we're going to introduce blood type, because that also happens to be something in a monohybrid cross. So we draw that. Whoops. What happened to draw? Okay. Anyway. Have a monohybrid cross here. And blood type is... The traits are determined by eyes. But here's the thing. There's a catch. Blood type is different. It has multiple dominant alleles. So you have I, which is dominant, and I, which is recessive. Recessive blood type, the only recessive blood type out there is type O, which I'll show you right here. Type O, blood type O. And meaning it doesn't occur often. It's pretty rare. And so you have that. But the thing about blood type is there's multiple dominant ones. There's a type A and a type B. So you probably know that these are 3 blood types, A, B, and O. And you're like, uh-oh, there's 2 dominant ones. What do I do? Well, I'll show you. So basically, what you end up having is you split these into 2, and you end up with an I with a little a in the corner, and an I with a little b in the corner. That won't draw. Anyway, and meaning you now have 3 alleles, which makes it so much harder. And there's a code, I think it's incomplete. Oh, geez. AB blood type, I believe, is codominance because they both show up. So all in existence, there are 4 blood types. I should have elaborated on this earlier. 4 blood types. Type A, type B, type O, and type AB. AB is codominant. And so, usually, you have a parent with, let's say, you have 2 parents with one of them has... I'm going to type this out. Actually, no, I can't. Yeah, I will. One of them has parent A is heterozygous for A, type A. And you can pretty much assume that meaning they have the dominant A and the recessive O, because that's what heterozygous is. Parent B, on the other hand, has homozygous, but they're type O. And you might realize, or remember, that O is a negative allele, meaning that it has to be homozygous. There's two recessive alleles. And so I'm going to set up this Punnett square for you, meaning you'll have, I'm going to show the answer after this, so you might want to pause the video before. If you want to do it yourself, pause the video now, because I'm going to go ahead and set up the Punnett square for you. Anyway, heterozygous type A, you have, realize type A is dominant, and so you have that, meaning type A. But heterozygous also means the alleles aren't the same, which means you'll have a little I right here, because that's recessive. A person with that genotype would still have type A blood, because the dominant, as always, the dominant trait masks the allele trait. What? The dominant allele always masks the recessive allele. Parent B, homozygous type O, meaning negative. They have recessive. I can't talk. They have both recessive traits, meaning a little I and a little I. I can't dot the I. Anyway, you know their name. And I'm going to write it out for you, so if you don't want the answer, I suggest you pause the video right here. And anyway, you end up with, when you cross it, you have IA, or capital dominant, and lowercase a. And then you do the exact same thing, and you cross it over. Do it again. It's like Battleship. If you've played Battleship, you know exactly how to do this. And you end up with that. And so there's 4 possibilities of the offspring of these parents A and B having a certain blood type. And now you know that 2 out of 4 times for example, if they were to have 4 children, chances are 2 of the children have type A blood, but are heterozygous for type A. And 2 children are homozygous, recessive, meaning they have type O blood. And that heterozygous type A is something I'd like to address. There's this word, I know this is a mess, but there's this word, carrier. And it means that while you exhibit a certain trait, while you have a dominant trait that masks that recessive, you still have that negative trait. So you're a carrier for type O. So this child in the top left, like this child for example, this child is a carrier for type O blood because they have the recessive gene, but it just doesn't show. And so that's what a carrier is. I should have explained that earlier. But that's the end of this very confusing tutorial class lesson. And this was just the basics of genetics. Hope you learned something. And just thank you for watching. Bye.